Hello everyone, this is the second piece for the cell lecture. We've already talked about the plasma membrane and the appendages of the plasma membrane, and now we're going to look at the cytoplasm and the nucleus, so the stuff inside of the cell. For each of those organelles, I'm trying to be very clear about what I expect from you. You need to be able to recognize it by picture. Okay, so if I show you a picture of something or show you a picture or a model, make sure that you should be able to recognize them. You also should be able to recognize them by a description. So if I try to describe them with words, you should be able to figure out what I'm talking about. And you should know the general function for each of those organelles. Okay, so let's talk briefly about what is an organelle. Okay, so organelle, if you look at and you break that apart into its roots, it's defined as a little organ. And so really what it is are structures found inside of cells that are membrane bound. Okay, so it has the exact same phospholipid bilayer that we already saw with the plasma membrane, and they have a particular function. So it's just the structures inside of the cell that do something for the cell. Okay, so let's look at the nucleus first. Now, technically, the nucleus is not an organelle, but you still need to know that structure. So if you're looking at this picture here, usually the biggest structure inside of that cell is going to be the nucleus. And the nucleus is very important because it contains your genetic information, your DNA. Okay, so it's going to house that DNA and protect the blueprints of life. And around the outside of the nucleus, you have what's called a nuclear envelope. The nuclear envelope just creates the external boundary of the nucleus. And embedded in that nuclear envelope, you have a bunch of pores. And the purpose of those pores is to allow something kind of specific to enter and exit the nucleus, and that's going to be messenger RNA. So for those of you who have ever learned about protein synthesis, that would be how those messenger RNAs exit the cell. If you don't know what I'm talking about, that's okay. That's not really part of this class. It's just a side note. Okay, so let's go into the next one is the endoplasmic reticulum. Endoplasmic reticulum. Okay, so one thing that I'd like to point out is that the endoplasmic reticulum actually has two different organelles that are both endoplasmic reticulum. You have the smooth ER or smooth endoplasmic reticulum and the rough endoplasmic reticulum. So this would be the rough, this would be the smooth. What's different about them? Where does their name come from? And it has to do with the presence or absence of these little pink dots, which are called ribosomes. When you look at the rough endoplasmic reticulum under the microscope, it's got this little studded appearance that makes it look rough and grainy, and that's because it has ribosomes. Now, the function of those ribosomes is protein synthesis, which just means decoding that messenger RNA to create proteins, make proteins. So the word synthesize means to make, so we're just making proteins. And those ribosomes are found two places. The first is just floating freely in the cytosol, just sort of hanging out in the liquid. And then the second place they were found is attached to the outside of the rough endoplasmic reticulum. Okay, so the rough ER. And so if the function of the ribosomes is to make proteins, what's the function of the rough ER? You know it already. Make proteins, same thing. Okay, the other one is the smooth endoplasmic reticulum. It does not have those ribosomes, and therefore it is not making proteins. It is involved in creating things like steroids and some certain types of enzymes, but I'm going to focus on that one less. Okay, I definitely need to make sure you understand the rough endoplasmic reticulum more so. Okay, so ribosomes, they make proteins, protein synthesis. Next one is the Golgi Complex, also known as the Golgi apparatus. Sometimes I say that kind of flip-flop both, but I think your lab checklist says Golgi Complex. So I'm going to try and stick with that one. So the Golgi Complex, basically what we're looking at here is if you take a bunch of phospholipid bilayers, the same phospholipid bilayer we saw with the plasma membrane, and it just creates these sort of flattened stacks of that phospholipid bilayer, of those membranes. And you can see, if you look carefully, at the very ends of those stacks of membrane, you have these little bulbous ends, these little rounded edges. And the reason they're bulbous like that is because those are going to, so if it's like this, those are going to pinch off and create what are called vesicles. And the function of those vesicles is to move up to the surface of the plasma membrane. It'll fuse with the plasma membrane because they're both made of a phospholipid bilayer. And that'll allow anything that's inside of that vesicle to be secreted or spit out of the cell. That's called exocytosis. Okay, so the function of the Golgi apparatus would be to package and export any of the proteins. So those would be things like hormones. For example, if you're looking at a gland, an endocrine gland, the product could be a hormone or something like that. Okay, so this would be how you get it out of the cell. You use the Golgi apparatus to do exocytosis, which removes and makes the cell, makes the whatever's inside go out, like the hormone, for example. Okay, so what I do want to take a minute is let's pause, because if you look at the endoplasmic reticulum and you look at the Golgi complex, at first glance, they look awfully similar. Both of them are stacks of membrane. So let's look at some strategies for figuring out, well, how are they different? One strategy is that because of those vesicles, sorry, I don't mean moving all over. Because of those vesicles at the ends, the Golgi complex will always be shown, even on models, with that nice bulbous end, okay? The other thing to look at is the location in the cell. The endoplasmic reticulum, if you look carefully here, the boundary of the endoplasmic reticulum, that membrane that makes up the endoplasmic reticulum here, is continuous with the nuclear envelope of the nucleus. So because they're continuous with each other, the endoplasmic reticulum is usually shown stuck right on the nucleus. Okay, so you'll have the endoplasmic reticulum nice and continuous and sort of stuck on the nucleus, whereas the Golgi apparatus with its little vesicles budding off will be kind of floating further away. Now, I did notice that one of the model pictures that I've seen has the Golgi complex attached for some reason. So be careful, that's not usually the case. Okay. Let's go into lysosome. So when we see lyse, what does it mean to lyse something? It means to break it apart, okay? To put it apart. And so what a lysosome is, it's a little membrane-bound sac. And I like to think of it as sort of like little bubbles, right? So you got these little membrane-bound bubble inside of the cell. And inside of that lysosome are digestive enzymes. And digestive enzymes, their function is to break things apart, okay? So what the function of the lysosome is to fuse with other types of vacuoles, for example, like a food vacuole, if something has been brought into the cell, or maybe a damaged organelle that needs to be recycled. And then it'll release those digestive enzymes, and then the digestive enzymes will break it apart, and then you get to use those little building blocks for other parts of the cell. Okay, so I really like to think of the lysosomes as the stomach of the cell, because that's really what it does. It digests things so that you can reabsorb it and make it into something else. Okay, so you can move it and use it for other structures within the cell. Okay, so lyse means break apart. Mitochondria. So mitochondria, they're called the powerhouse of the cell, and they are important for creating ATP, which is just a form of energy that the cell can use to get things done inside of the cells, things like moving things around or things like that. So anytime the cell needs more energy, it'll come from most of it comes from the mitochondria. Now, when you look at the mitochondria, you should always be able to tell it's a mitochondria because unlike all the other organelles, it's a double membrane. It has two phospholipid bilayers, so two double membranes connected to each other. Okay, so you have the outer membrane that'll be circular, kind of around the outside. And then you have an inner membrane, and this inner membrane is going to be very highly folded, okay? So anytime you see a picture of a mitochondria, I like to think of it as it looks like it has stripes. If it looks like it has those internal stripes like that, that is going to be the mitochondria. And that inner membrane are going to be folded into what are called cristae. Okay, so those inner membrane that form the cristae create that striped appearance, and there you go, you got a mitochondria. Okay, so I want to quickly talk about, well, why is the inner membrane highly folded? Okay, so think about it. Why did we see those microtubules be highly folded? Why do you have extensions like that? The purpose is always to create more surface area. And the reason for that is that the machinery that is used to make that ATP, to make that energy, is embedded in this inner membrane. And the more membrane you got, the more energy you can make. So double membrane, striped appearance. Okay, so what I would like you to do now is to go ahead and pause the video and see if you could draw me a cell. Okay, so I want you to draw me a cell and then label all of these things. I will not be collecting this, but I still think it's worth your time. So I'm going to draw you one first, but I'll make a separate video.